Alright guys, so now that we've taken a look at sort of an overview of cellular respiration and an overview of glycolysis, which happens in both anaerobic and aerobic respiration, we're going to take a look further into aerobic cellular respiration. So this is cellular respiration in the presence of oxygen, and this occurs in like breathing organisms, organisms that use the oxygen in the air to respire. So last class in the last presentation here, we took a look at glycolysis. So the process on the right in the image is the physical process of glycolysis breaking down. So the breaking down of this glucose molecule, you'll see here at the top. Okay, it's a 6-carbon sugar. You'll see 1, 2, 3, 4, 5, and 6. All right, and then we see here we have energy investment. Okay, we have 2 ATP. Energy is invested here. We've used ATP, and we have some high energy molecules pulling hydrogen out of this reaction, and then we get a net or a gross of 4 ATP produced with only a net of 2 ATP produced because we have energy investment here and energy payoff. Okay, you'll see then we create 3 pyruvate, 3 carbons, pyruvate here, and there's 2 of them. So there's our 6 carbons here, matching our 6 carbons at the top. So as I mentioned, what it is, it's sugar splitting, and it's broken down into 2 stages, the first one being energy investment with energy payoff. Okay, and this, remember, occurs in both aerobic and anaerobic respiration. So just a quick review on what the mitochondria looks like. Here's the outer membrane, the inner membrane here with all the folds, okay? All these folds are called cristae, and inside these cristae we have a fluid that is called the mitochondrial matrix. Okay, in between the both membranes is called the intermembrane space. So we have the outer membrane, the inner membrane, and in between that space, okay, filling the outside of the folds, is called the intermembrane space. All right, so if we're looking at, again, sort of an overview of cellular respiration, this whole blue thing would be the cell. Okay, we've already looked at glycolysis in the cytoplasm, so we know that this occurs outside of the mitochondria. And this is an oversized mitochondria, just so we can look at the processes within that organelle. So we see that we've already looked at step 1, and right now we're going to take a look at step 2, 3, and 4. So the first step here is pyruvate oxidation, and we'll get into talking about what that is in just a second here. And we see that there's some product that goes into this cycle called the Krebs cycle. So Krebs cycle is step number 3. And what you see here is that there's NADH produced throughout the Krebs cycle, as well as an FADH2. Okay, and this is a process called oxidative phosphorylation. But essentially we're just creating really high energy molecules. All right, and same thing as in the photosynthesis electron transport chain, these get produced and sent to the inner membrane of the mitochondria. Okay, and as you'll see in the inner membrane of the mitochondria, we have a variety of proteins. Okay, these proteins transport electrons through the electron transport chain, and at the end we have this special protein called ATP synthase. And the way I know it's ATP synthase is we have a product of ATP here, and it's at the end of our electron transport chain. Okay, so let's take a look at these next 3 steps here and how they work in aerobic cellular respiration. Okay, so as I mentioned, we've looked at all of these steps sort of individually on that overview, and we're going to look at these last 3 in more depth right now. So step number 2, so after we break glucose down into 2 pyruvate molecules, we need to look at how these molecules are then further processed. So pyruvate oxidation is a chemical pathway. So what this means is that there's a variety of different enzymes or proteins that help this reaction occur. So we have glycolysis in the cytoplasm, and we have the Krebs cycle in the mitochondrial matrix. So this process of pyruvate oxidation connects both of these processes together. And the process begins when two pyruvate molecules are transported from the cytoplasm into the mitochondrial matrix. So that's kind of how the process begins. And three changes occur. Okay, a carbon dioxide is removed from each pyruvate molecule. So a carbon dioxide is pulled off from that simple sugar molecule. The remaining two carbon portions are oxidized by NAD positive. Now remember, oxidized means it's losing electrons, okay? And it's losing electrons through this high energy molecule called NAD positive. Each NAD positive gains 2 hydrogen ions, which means that it's 2 protons and 2 electrons. So this is where it's saying that our carbon portions are losing electrons, okay, as they're being oxidized, and the electrons are getting transported to the NAD positive molecule. Okay, the remaining 2 carbon compound becomes an acetyl group. So an acetyl group, if you're in chemistry, you might know that an acetyl group is just a specialized functional group that we can kind of attach onto the end of molecules. And this isn't super important. You just need to know that it's a 2 carbon compound, and it can kind of be attached to other molecules. Okay, a compound called coenzyme A. So this is our enzyme here that I was mentioning, becomes attached to the acetic acid group, forming acetyl-CoA. So we combine this 2 carbon compound here with coenzyme A. Okay, and this coenzyme A and this carbon group become acetyl-CoA. Okay, this molecule is then transported to the Krebs cycle, and this is sort of where this molecule gets broken down further. So this process of pyruvate oxidation is just sort of a processing phase. So here's what it would look like. We have pyruvate here at the top. So this is our product of glycolysis. And our product of glycolysis is, remember I said that a carbon dioxide is removed from the pyruvate. So we have 1 carbon atom here. And then it attaches with coenzyme A. So what happens is coenzyme A comes in and it's oxidized by NAD positive, producing NADH. So remember that 2 protons and 2 electrons are removed here, and this molecule here becomes our electron carrier. All right, we have coenzyme come in, and we have 1 carbon removed, and then we have a product here of acetyl-CoA. So you'll notice that the coenzyme A is attached to the side of our 2 carbon compound. Okay, this NADH is then used later in our electron transport chain, which you will see, and then this acetyl-CoA is transported to the next step, which is called the Krebs cycle. All right, so 2 molecules of acetyl-CoA enter the Krebs cycle. I say 2 here because per every pyruvate, we have 1 acetyl-CoA produced. So because glucose produces 2 pyruvate, it then therefore produces 2 acetyl-CoA into the Krebs cycle. 2 molecules of NADH proceed to stage 4, which is the electron transport chain, as I mentioned. And the 2 carbon dioxide molecules diffuse out of the mitochondria and out of the cell as waste. So this is where we have carbon dioxide produced, is in this pyruvate oxidation. This is important, again, to know the overall net reaction of cellular respiration, where carbon dioxide is actually produced. Okay, so let's take a look at this Krebs cycle here. The Krebs cycle is just a series of reactions that converts these organic molecules into these higher energy molecules that are usable forms of energy. So we convert our organic, for example, our acetyl-CoA molecules into ATP, NADH, FADH2, and we also remove some carbon atoms as carbon dioxide. It's an 8-step process with many enzymes involved, and again here, just like the Calvin cycle and photosynthesis, I don't expect you to know all the intermediates. I think that's quite tedious to know, so I'll highlight what is important for you to get out of the Krebs cycle here. Alright, so as we take a look at this image here, it looks quite complex, but don't stress it because this part you already know. This is pyruvate oxidation here at the top. We have a 3-carbon pyruvate molecule, again attaching with acetyl-CoA. A carbon dioxide is released, and oxidation by NAD positive, removing electrons and protons, creating this electron carrier, which then gets sent to step 4. Okay, now we're looking at this cycle down here in purple. So acetyl-CoA is this molecule at the top here. It's a 2-carbon sugar with our coenzyme group attached. And what happens is acetyl-CoA gets broken off into the mitochondrial matrix. So it might seem a little bit redundant to have this enzyme added and then immediately taken off, but the enzyme is actually needed in order for transport into the mitochondrial matrix. Without that sort of special marker, it doesn't happen. So what happens is this 2-carbon compound, okay, is then broken down and 2 carbon dioxide are produced. So we have a further release of carbon dioxide molecules, okay, and this again gets transported out of the cell and as a waste product, which you then breathe out. So this would happen, for example, in muscle cell of your body, and through your circulatory system gets sent to the lungs and you breathe out and it's expelled. All right, we have further oxidation here. We have NAD positive molecules producing NADH and a 2 hydrogen group. Okay, so we have more electron carriers produced. Then we have the formation of some ATP. Okay, so we have an ADP and a phosphate group producing 1 ATP molecule. We have again oxidation here with the FAD and FADH2 molecules, and again further oxidation producing another high energy molecule here, NADH. So you'll notice that in the Krebs cycle we have a total of 3 NADH molecules produced and 1 FADH2 molecule produced, carrying our high energy electrons, which then get transported into the electron transport chain used to generate ATP. All right, so the Krebs cycle occurs 2 times for every glucose molecule. So what I mean by this is we need 2 rotations of the Krebs cycle, 1 for each pyruvate or for each acetyl-CoA molecule. Okay, the acetyl-CoA enters the cycle and the CoA is released and can be used for the next pyruvate. So this is recycled. In one complete cycle, the total of 3 NAD positive and 1 FAD are reduced from 3 NADH and 1 FADH2. So I've mentioned that in the previous slide with the image there. In one complete cycle, an ADP and a phosphate group are combined to form 1 ATP. So again here, we only have 1 ATP produced. That is not enough for our everyday functioning. So we need to see where more ATP is produced here. And again, 2 carbon dioxide molecules are produced and expelled in the body as waste. So here, if you are really interested in all of the intermediates in the Krebs cycle, we can go through them here. I don't think it's super necessary. You'll see the important steps. Acetyl-CoA goes into the cycle. We have carbon dioxide produced, NADH produced twice here. Okay, so this is the 2 NADH and the 2 carbon dioxide produced. We have oxidation through this NAD positive molecule coming in. Here's our ATP production, our FADH, and our last NADH molecule produced. And you'll see here coenzyme is recycled and attached over here in our pyruvate oxidation. Okay, so stage 4. Remember what chemiosmosis is. So we talked about chemiosmosis in photosynthesis, and osmosis is simply just the transport of molecules down a concentration gradient, and in this case we're looking at the concentration of hydrogen ions down a concentration gradient. So what we're looking at is the production of ATP through the electron transport chain in the inner mitochondrial membrane. So oxygen is our final electron acceptor. So what that means is our final electron goes and attaches to this oxygen atom, and it's our final electron acceptor, and this is a continuous process. The hydrogen ion gradient drives ATP synthase. So as you see here, hydrogen ions move through these through the membrane here, and we have again the formation of water. So this is the opposite of what you would see in photolysis, and this is in intermitochondrial membrane. Again, hydrogen ion concentration up this way, and then through this specialized protein called ATP synthase, we have the formation of ATP, as you remember from photosynthesis.